Welcome and thank you for attending today's webinar. My name is John McKinley and I will be your host today. If you experience difficulty with audio or advancing slides, please refresh your browser window. Today, we have a presentation on a collaboration between Cartier and the University of Twente. Leon Terstappen, a professor at the University of Twente, will present on a new multiparameter assay for the immunity monitoring of COVID-19 patients. Leon is a medical doctor with a PhD in biophysics who is intrigued by finding technology solutions to medical problems. He is an internationally recognized expert in cytometry and the detection of rare cells. First, Judicael Perisseau, our senior application scientist in Europe, will introduce the Cartier LSA technology used in the immunity monitoring assay. Before working at Cartier, Judicael worked for 10 years as a scientist in the pharmaceutical industry at MorphoSys, where as part of the interaction analysis group, he was involved in the discovery and development phase of therapeutic antibodies. Judicael holds a PhD in chemistry with a specialization in structure analytics. After today's presentation, Richard Shashford from the University of Twente will join us for a panel discussion. Please submit your questions with the box on the right side of the screen. I'll hand it over to you now, Judicael.
Thanks, John, and thank you for everyone who was on the call. So today I'm going to give you an introduction about the Cartier LSA platform and some details on how the system works. And after my presentation, you will hear from Leon about an application using the system for medical diagnostics. When it comes to antibody discovery, there are awesome tools out there to generate high-quality antibodies in a very elegant way. And characterization of this antibody is a necessity, and there's no point in generating all these high-quality potential therapeutic and diagnostics if there's no way to leverage them. And pretty often, we see just due to the lack of ability to handle them in terms of throughput, we sort of discard quality candidates. So to meet this need is the Cartier LSA platform. This is a novel system based on SPR detection technology. And what is new about it is the fact that it has a 3D configuration allowing 96-channel multi-printing or single-channel injection across the array. So the instrument has a dynamic with itself that creates an array of 384 ligands on the surface of a biosensor, and it can flow one injection that crosses the array. So we're monitoring a total of 384 signals per injection, non-including reference signals. And now I give you guys a little synopsis here on how each side of the instrument works. So the multi-channel mode on the left-hand side of your screen. So now you can see the sampler drawing 96 samples at a time, and then our proprietary printhead technology flowing this sample on the top of the chip surface to create a ligand array. Then, in a second step, our single channel docks across this array, as that was just built, and inject an analyte across it to measure real-time binding using minimal amount of samples to do so. So you will see later that in Leon's presentation, actually, that the printhead, which is typically used to generate ligand array in early discovery, as shown here, can be also used for the characterization of sample. And in the case of the collaboration with University of Twente, we use it to characterize serum sample. So a maximum of 384 serum sample has been characterized in one single experiment. So now I jump into the assays themselves, so not the assays that Leon will present later, but the kinetic assay to measure real-time binding kinetics of typically antibodies and antigen. In this exercise, up to 1,152 antibodies can be screened in a single experiment using 384 well plates of antibodies. So the antibodies themselves are captured on the surface via the Fc part. Then we come with the antigen titration to measure the kinetic rate constants. So the advantage of this assay is that it's highly parallel. So we measure in a single injection the interaction of the antigen with 384 antibodies simultaneously. So there's no disparity of changes of the quality of the antigen over time. Also, the array capacity allows, if there's no 384 unique clones, to build in replicate, which is until now not performed in SPR at this stage of a project, so in early discovery. So the LSA changes the data quality you can get early on, and the same array capacity can be leveraged to titrate the ligand on the surface, so trying different surface densities within a single experiment. Then choosing the best data ultimately for kinetic analysis. So it's something, one more time, it's something that's never been done historically, as most biosensors require a day or two of assay optimization, then running the actual experiment. So optimization in the case of LSA is a one and done process. And really, that's what we are chasing in the end, high throughput kinetics. So here are shown 384 ligand kinetics, and within each individual type is 8 analyte concentration titration. So all is done in a single run. And the picture is actually a screenshot of our software, and the software is able to flag data that may be borderline. So in gray are shown data with a low signal, in a yellow data with a complex signal. So to generate a 384 data set like shown here, it takes about 6 hours. So this is quite fast to obtain 384 affinities measurements. And I skimmed over it previously, but there is the ability to build in replicate into your assay, and we found a really good reproducibility on the system. So in this case, I'm shown on this slide, we are looking at 12 replicates, the same antibody of the same ligand, and we're looking at the mean of rate constant calculated from these 12 replicate and the standard deviation from this replicate as well, giving high confidence in the determined kinetic parameters. So we have good agreement among the data just obtained from generated from this 12 replicate, showing that you can build, replicate, and get statistical confidence in your assay. And to build upon this, there is always a question if we have a good reproducibility within the assay and how this data translate outside this individual assay. So from an accuracy perspective, we can say they agree very well with the Biaco, for example, a well-established platform that most people just reference when looking at label-free binding. So there's been an excellent paper put out last year by folks at Adimab and Amgen looking at benchmarking different SPR platform. And the LSA was right in step of what is a gold standard of SPR, the Biaco, showing really good agreement with individual rate constant and affinities. So here are shown the affinity of 36 clones. And the difference is that the LSA is obviously faster and require much less sample and can finish this type of assay in a day versus multiple days and run to be achieved on other platforms. So only you have speed, but also you have confidence in the data you are generated. And I will add that in this assay we measure this 36 clone in replicate since we have the capacity to do so. With that, I shift to epitope binding, which is the other main application with kinetics on the LSA. So epitope binding is a pairwise exercise where we take antibody and compete them with each other to determine if they can form a 3-molecular complex with the antigen, and the goal of this is to group antibodies is what is called beams or community. So it's very, very advantageous to perform epitope binding in early discovery in order to identify diversity in the set of samples you characterize, and to maintain that diversity going forward as candidates progress in the therapeutic discovery. So there is so much information from this data that changes the mindset of drug discovery, where historically it's kind of funnel process where we try to get rid of the high number of samples to have a number of samples that is manageable. And now we are looking at a sampling strategy using epitope details. So a nice thing about epitope binding as well is it allows to have details about a sandwiching pair. And that sounds very well for diagnostic and also assay development. So the main user requirement for epitope binding, like for Kinetics in early discovery, is low sample consumption and throughput that's completely transformative. As I mentioned previously, the LSA can array 384 ligands on the surface of the chip, and then in a typical binding experiment, we will bring an injection of the antigen in each cycle, then an injection of one of these 384 antibody. So for each injection, we measure competition against 384 species. And in total, we generate 147,000 interaction measured in a single experiment. It's a huge number of data points and details about the candidates. Great things about it, obviously, is the low antibody requirement or consumption that fit very well with early discovery, where the amount of material is pretty limited. And even the antigen level are very low, considering we are under 200 microgram of antigen used for an epitope binding in its largest format, so 384 by 384. And at the back end of it, we have our proprietary software package, which is very interesting, integrating visualization tools that really allows you to take these huge data sets and make meaningful inference of them. And that's something that's tangible for an investigator to understand. So the other exciting things in the epitope binding software is the ability to pull in all the antibody attributes onto our proprietary network plot. What I show here is from a publication of past year, and we can see different community network for antibodies, and each antibody is represented a kind of node on this plot, and the line between antibody are blocking relationships, so indicating just antibody overlap. So we can see we have very discrete communities. We do not have any communication with each other. So what we can also do with this data in the software is bringing piece of information that describes this antibody, but still in the context of epitope. So for example, library source, expression level, affinity, cross-reactivity. There's no limit how you can layer data on the top of this network plot. So it is a hugely powerful tool that for not only to visualize the epitope landscape, but also to visualize attributes of this antibody that are meaningful for their selection criteria. So that was my introduction to the LSA system and its major application, epitope binding and kinetics. I'd like to switch gear a little bit, and before to hand back over to Leon, I would like to quickly present the status of Cartier in the fight against COVID-19. So we have a lot of customers that leverage the LSA to meet the needs of COVID-19 research, and they are listed here on the right of this slide. So there are academic and public institutions that are involved in this fight, but we have also industry customers like Eli Lilly or Twist. Additionally, the LSA has been selected by the Gates Foundation that put together an antibody consortium screening arrangement where the device will be a key platform to perform epitope binding and kinetic analysis to select potential therapeutics. So that really encourages us at Cartier that can contribute to this effort and be part of the solution. And in order actually just to accelerate the different discovery workflow, we developed and now offer a biosensor pre-functionalized with the receptor binding domain of the spike protein, and this chip will carry the RBD protein on its surface, and it is a ready solution to Characterize and anti-RBD antibodies. So now the last thing I would like to touch is a kind of an expansion of the LSA application, which is a serum-based assay. So I don't want to say much about this assay, but Leon will. But this assay will highlight that the LSA platform is not only limited to discovery through the selection of therapeutics, but could be potentially used for medical diagnostics. And now I hand back over to Leon, who is going to go ahead and show you nice looking data generating during the development of Assay to monitor human response to infectious agent. Thank you.
Welcome to this webinar. I am Professor Leon ter Stappe, chairholder of the Medical Cell Biophysics Group of the University of Twente. I will present this webinar for Cartier on behalf of our COVID-19 team. At the time the Netherlands went into a COVID-19 lockdown earlier this year, Richard Schasvoort from our group moved our SPR equipment to the local hospital to start the development of an assay to profile the immunity for COVID-19. Here we will present the results. Here we show the COVID-19 virus. The two important immunogenic proteins are the nucleocapsid protein and the spike protein. The spike protein contains the important receptor binding domain, or RBD. And on the bottom you'll see that the SARS-CoV that appeared in 2002 and the SARS-CoV-2 that appeared in 2019 are quite similar. Here you can see the amino acids sequence of the spike protein of the coronavirus. It contains the S1 and the S2 subunits. In the S1 subunit, the receptor binding domain RBD is located. The RBD binds to the antigen tensin converting enzyme 2, or ACE2, of cells. It is important that the virus is blocked by neutralizing antibodies to prevent binding to the ACE2. To protect and cure COVID-19, an active immunization with a vaccine that generates neutralizing antibodies is needed, as well as a passive immunization strategy to diseased COVID-19 patients to treat with anti-COVID-19 antibodies. For COVID-19 immunity profiling, it is important to measure the total immune response of the patient. The patient generates the isotypes IgM, IgA and IgG, and it's essential to find out how the polyclonal response binds to the various regions, such as the nucleocapsid, the S1, the S2 and the RB2. The affinity of the antibodies for their target likely is important for an adequate response to the COVID-19 infection and might provide important insights for the severity of the disease of COVID-19 infected patients. So, an hypothesis is, do we get long-lasting higher antibody response and strong antibody affinity, or a lower antibody response and weak antibody binding? How do we perform patient sample spotting on the SPRI sensor surface? The sample of corona patients are flown over the sensor surface with a corona protein immobilized on it. Only the COVID-19 specific IgM, IgG and IgA antibodies will bind to the surface. The sensorgram shows an example of the IgM, IgG and IgA antibody detection by injecting respectively anti-IgM in the beginning, followed by anti-IgG, followed by anti-IgA. And you can see here clearly the sequence of responses. So the principle of detection of the IgM and IgE and IgA concentration of COVID-19 patients is shown on the slides. So first we start with the sensor prism is coupled with the receptor binding domain of the SARS-CoV-2 to the sensor prism. Then the sera of 96 patients is spotted to the sensor for 15 minutes. Then we place the spotted sensor in the SPRI imager. Then we inject affinity purified IgM, followed by anti-IgG and IgA. So each interaction is 5 minutes, and the total analysis time is 26 minutes. And then we calculate the R values for each patient, as shown in these pictures. This slide shows a tile view of all 96 serial plots. So to illustrate the different observed responses of the various ratios of IgM, IgG and IgA, we highlight a couple of examples. So for all patients, we can measure this 3 isotype conjugate per patient simultaneously. So here you see no response, then you see a slight response, and then a little bit more. So this patient is completely different, so you don't see something in the beginning, but then a very rapid response. Then this one is again different. Here you see again difference, and here you see another difference. So what you already can see here is that the responses of the patients are quite different, and you can actually cluster them in different groups of responses. And of course, this measuring in time will become very important. Here we plot for all 96 patients the IgG, IgA and IgM and the total antibody response. We have, however, also want to measure the affinity. When an offline printer is used, then we do not know how much was bound during the printing process. This is needed to measure the on and off rate of the antibodies of the patients, which we feel could be of importance. So how do we perform affinity profiling? Fortunately, we can follow the binding process in Cartier LSA. It turned out that the on rate of the mixture of isotypes cannot be accurately measured due to the enormous mixture and concentration of polyclonal antibodies. However, the total response can be measured in the ELISA for an accurate off rate determination. And you see here the on rate and the off rate of the samples. So to perform these experiments, Richard Scharsford traveled to München, where they had an LSA machine, and here he is standing next to the LSA. And on the screen, the IgM and IgG and IgA measured performed on the LSA is shown. So how to get the data for the strengths of binding? So during the association process, we measure the response R in resonance units. In the dissociation phase, we measure the slope, the DRUs. And then the off rates constants can be calculated simply by dividing the slope with R. We applied an additional trick by adding the RBD to the dissociation buffer to prevent rebinding and getting a pure off rate for all sera at once. By zeroing the transition between the association and the dissociation phase, the slope of dissociating molecules can be measured. The weaker the binding, the faster the off rate. So if there is maturation in the immune response, then the slope for longitudinal samples will go up. The response in the association phase is already measured, and the LSA generates the off rate constants, KDs. When we plot the off rate as a function of the days after symptoms onset for all the patients, we observe relative large mixtures of all kinds of antibody responses. But clearly we see maturation effects that the off rate is going down. So, the strength of binding is increasing during the development of the disease. This was an important conclusion which is shown in our pre-printed paper available at the Research Square pre-printed platform. And of course you can already see here that we can measure in time and see clear differences in the immune response. What it means, of course, will have to be determined with more patients and more longitudinal studies. So in conclusion, we developed a rapid test for profiling COVID-19 immunity for 96 times 4 patients simultaneously. We can measure three isotypes, the IgM, IgG, and IgA, and the off rates of the anti-RBD antibodies for strength of binding. Maturation of the IgG response is shown in convalescent patients, and research is progressing. And with that, I want to conclude. So the results I've shown you was initiated by Richard Schaskort at the University of Twente, who brought together members of the University of Twente, Saxion, Cartier, Vicens, and MST Medlon, which is basically the hospital and the pathology department, to form our Twente COVID-19 team. Hope this was informative, and Richard Schaskort of the University of Twente can answer any questions. Thank you.
Thank you, Leon and Judical. We will now start the Q&A discussion. Please submit your questions with the box on the right side of the screen. There's a link to the preprint of the paper we presented on today at the bottom of this slide.
Judical, can crude samples be used on the LSA?
Thanks, John. My short response will be yes, they can use on the LSA. Actually, the LSA has been developed just to bring SPR in early discovery, so we don't expect all our customers just bring some purified sample. And actually, we have a number of customers using the LSA for primary screening, using, for example, bacterial or periplasmic extract to screen a single channel fragment. And some are using the anti-V5 tag just to capture this single scFv. It's a kind of ideal setup because the anti-V5 tag have a very high affinity for the tag, and the LSA is able also just to cycle the sample just during the capture of the scFv or just any ligand on the surface for a very long time. So even if you have just a limited amount of extract or low expressing clone, by increasing the contact time between your sample and the anti-V5 surface, you can easily just capture, enrich the surface with this ligand, and then get just a full kinetic characterization of your clone. And actually, the same is true for hybridoma. And just recently did some screening campaign for one of our customer using for COVID using hybridoma, and we also use some culture supernatant. So there's no limit in using a crude sample with the LSA. We just recommend, since we have a microfluidic system, so we recommend just to filtrate the sample, or just simply just to centrifuge them before just to apply them on the device.
Thank you, Judical. Yeah. We have a question about binning. What is the total amount of antigen required per clone, and what is the maximum binding affinity that can be measured with the LSA?
So to answer first the second question, what's the binding affinity, the maximum? So we are basically in the same range of affinity on the other platforms. So I'm not just give you a KD, but in terms of rate of association, we just can measure up to 10 to the 8. And in terms of on K-off, we are 10 to the minus 5 to 0.5. That is to give you just an idea in terms of just a kinetic parameter we can measure on the device. Now, for epitope binding, it will depend on the affinity of your antibody. But if you're just thinking running classical sandwich binding in a large format, so 384 by 384. Just to make some calculation, just a standard condition would be just so in such a classical sandwich binding, we inject for each cycle, we inject one concentration of one injection of antigen, which is followed by one injection of the 384 antibody you would like to characterize. So meaning you have about 384 injection of antigen plus of course some reference injection. So let's say 400 injection of antigen, and typically we work at an antigen concentration of 1 to 200 nanomolar. 200 nanomolar solution. So you can calculate just 400 injection with 200 nanomolar of antigen will give you an idea what the consumption of antigen you will need. But I can say we just performed a large binning with some antigen just staying under 200 microgram of antigen.
Thank you, Judical. Richard, what is the off rate of samples in the absence of RBD?
Yeah, that's a good question, because that's the trick we applied for better off rate determination. So perhaps it's even be better if we could optimize the RBD concentration. But the nice thing of measuring 384 off rates, only you spoil I think 200 microliters, I think, Judical, for having this off rate, but the concentration of the RBD should be optimized for the real 100% rebinding to prevent rebinding event. But it is definitely a way of doing it, and sometimes it can be 25-30% has an effect on the off rate. It depends also definitely on the ligand density. If the patient spotting on the RBD surface, if it is highly dense, then the RBD is not, the rebinding effect is less affected by the RBD. That's a ligand density effect.
And just to add to Richard, the presence of RBD is necessary since we measure the dissociation of spaces which are multivalent, and we got the IgM which is decavalent, we got IgA which are tetravalent, IgG are bivalent. So just to avoid to have a huge rebinding effect, and just to be able just to discriminate the K-off and the different samples, the presence of RBD is necessary during the dissociation of the sample from the surface.
Yeah, definitely you will get a better parameter if you add RBD. And the whole assay is strength of binding is based on the off rate. There is also we found a good correlation, but then we tested patient by patient in different concentration, etc. That there is and then also you don't know the concentration, but then you reference the concentration, etc. But we found a good correlation between the affinity constant and the off rate. So now we can say strength of binding is correlated to the affinity or avidity, what you can say, because it consists of a lot of antibodies altogether who are binding to the full RBD, the IgM, IgA in a mixture spot. So that is really but we think the strength of binding is now very good. And the nice thing is that when you measure off rate, you are not depending on the concentration, because the concentration of your buffer is 0. You know the response on your spot, so it is concentration in there. How much it was captured, we observe then the off rate, and that's the nice thing. because in an on rate, yeah, you need to add a concentration. But what is the concentration and which one you should take, IgG, IgM, or whatever? So that's quite difficult. So this is an affinity parameter that's very effective to be measured, and also with this trick of RBD, you can measure this simply.
And actually, just for the characterization of different sera, we don't need just to have we don't need to have the KD or the K on. I think the K off is the most important parameter here, just defining the different the strength of the binding of each sera, as well as after the isotopization, which is allowed to determine the quantity of each species in the sera. Yeah.
Yeah, I agree.
Here's a question about detection anti-IgM, G, and A antibodies causing cross-linking, affecting the affinity measurement.
No, we are not measuring in that phase. We are not measuring the affinity of the binding of anti-IgM and then measure the off rate, etc. No, this is a polyclonal injection of anti-IgM and anti-IgG and anti-IgA. It's only for getting the ratios of concentrations what's bound from the serum. We measure the off rate earlier, directly after binding. So we are not measuring it in these first anti-IgM binding events.
Thank you, Richard. Judical,
yeah.
Can you perform kinetics and epitope binning assays on the same chip?
Yes, but there is a bit of nuance how you set up your assay. So particularly when you want to measure a kinetic in SPR, you would like to have just optimal density of ligand on the surface, just to avoid mass transport limitation, rebinding effect during the association phase and the dissociation phase. While when you're running epitope binning, you're not concerned about this artifact, which is mass transport limitation. What you want to see, epitope binning is a binary assay when you see additional signal or no additional signal. But actually, the two assays could be just combined on the same surface. We have a different customer just binding, running now on using our new Navigator software. We just installed recently by our customer, just allowing to queue different assay in the same to queue different method in the same assay, just running so first a kinetic on the surface and then epitope binning on the same chip. If you just have the same immobilization solution. So in principle, yes, you can just do both assay on the same surface, and which is great because you can have not only the affinity of the antibody for the antigen, but also where it does bind on the surface of the antigen. So there's plenty of information in two queued assay, and just you put all your sample on your device and you can walk away.
Thank you, Judical. We have one more question for Richard. Did you compare SPR with ELISA, lateral flow assay, or other techniques?
Yes, we compared isotype measurement as a concentration test, where SPR applied the RBD antigen, the ELISA, the S1 antigen, and a Roche platform, the COBAS, they applied a total nucleocapsid response. And as you can see in the preprint on the ResearchSquare platform, we showed the data and found some correlation with S1, and it's shown as quite scatter correlation with nucleocapsid measured by Roche. So first, people said your SPR instrument, etc., is not the same as Roche. So it's not a good. And I said no, the data of SPR is okay. So this was really remarkable that we could see such different levels that the protein part of the virus generates. And later we injected the serum of the single patient over various proteins, so a typical multiplex test, and we observe also a scattered level in SPR, so against S1, S2, RBD, nucleocapsids, variation per patient, different status of disease, etc. And I'm really excited how this response in COVID-19 does revolve. Why does a patient generate such a scattered immune response to COVID-19? And I think more subsidy for the university is necessary to find out this for COVID-19. So I think many years of research is needed before getting really the clinical relevant SPR test. And I think also academia and institutes and industry in Europe should cooperate, and perhaps also in the US on an international level, to solve the many scientific questions, because there are so many things you can ask for what really is the case with this patient, etc., and why it's binding and in relation to the angiotensin receptor binding, etc. And I am daydreaming about a breakthrough with where SPR, a unique technology, can generate the clinical data. So it tells something about the status of the patient. And then definitely we need then a further future vision. And my attitude is really optimistic, but as you may know, not any clinical test is based yet on SPR. So it costs years for getting such instrument or whatever. And the LSA, in my opinion, as a very early discovery tool, is needed for this kind of research. And I hope also that many, many more researchers all over the world will step into this patient immunity testing and see cooperating, finding epitope binding, etc. And this is my answer. And I thank also Cartier for getting me the opportunity to do this. I also am very with Leon. Leon did his presentation on the data, etc. But I'm and we will see each other, I think, hope in the future. Thank you.
And I also share the excitement of Richard just bringing SPR in medical diagnostics at the moment where we use it for early discovery. And just to give you an idea, of course, when you go to diagnostics, you think you need just speed and to have some assay which is pretty cheap. So just to give you an idea how long it takes just to analyze the 384 sera on the LSA, it takes between 3 and 3.5 hours. And I just to make some calculation, how much would just cost to the analysis of one single serum sample, and we just under $2 just per serum. So we just perfectly in line with the cost of diagnostics.
Yeah, I totally agree, Judical.
Well, thank you so much, Leon and Richard from the University of Twente and Judical from Cartera. And I especially want to thank you, the audience, for attending today's webinar. If you have any additional questions, please reach out to Richard or Judical with their email addresses listed on the screen. Thank you all for attending. I hope you have a great day.